Okay, well, we'll kick off the proceedings for today. Just like to welcome everyone to MND Connect Research Live, and thank you for taking the time in your weekend to dial in, both for our participants and also for our panel members. For those of you on Zoom, you may see a notification about the meeting being recorded. Please just click OK and agree to continue. First up, we'll have an acknowledgement of country. In the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Gadigal people on whose land I am today and pay my respects to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islanders peoples. So many of you have been dialing in to our State of Play sessions throughout the year. Today we have a bit of a different format. So we have a panel of 6 stellar presenters today covering a wide range of research expertise and roles. My co-moderator today is Peter Chambers, who is himself living with MND. And so with Peter having the most important perspective into the value of research for MND, he'll lead us off with his thoughts today. Next up, we'll have Julie Atkin from Macquarie University talking about how basic research approaches can drive treatment development and how her research contributes to this. Rebecca Ahmed from the Sydney University Brain and Mind Centre will discuss the critical role of biomarkers. Anthony Kari from the Perrin Institute in Perth will discuss the recent results from the Tofersen trial and also some other approaches he's looking at using gene therapy. Susan Mathers from Calvary Healthcare Bethlehem in Melbourne will discuss basically the treatment care continuum. And last but certainly not least is Beck Sheen, my counterpart at Fight MND, who will tell us about how research funders can help drive research forward. Each of our presenters will talk for about 10 to 15 minutes, and then we'll have a panel Q&A session following the presentations. So please submit your questions through the chat or Q&A function if you're on Zoom, or if you're on Facebook, send in your questions through the comment section. As we've got a wide range of expertise on tap here today, so please feel free to ask questions around pretty much any aspect of M&D research. We'll all do our best to address your questions, and what we'll do is we'll collate your questions if you fire them in as the talks are going on, and we'll present them to the panel at the end for the joint Q&A session rather than asking them throughout the session. So I think that's enough for me for now. So please, Peter, if you're ready, please take it away.
Terrific. Excuse me, that's a good start. Frog in the throat, sorry about that. Thank you, Gethin. I really appreciate the opportunity to chat. I suppose I should start with my story. I was diagnosed in late 2019, having battled with some symptoms for probably with hindsight 2 or 3 years. I had a surgery for a spinal fusion, which was hopeful of bettering the symptoms that I had, but that was unsuccessful. Long story short, sort of 18-month period transpired between the surgery and being diagnosed, and then it was confirmed for me in early 2020. And we all know what a terrific period we've had since 2020. So that compounded a little bit, but we move on. So my diagnosis, I've got a slow progression version of MND and flail arm. So in that sort of nearly 2-year period since being diagnosed, my arms now are pretty much not useless. It's too strong a word, but I require support and assistance for changing, dressing, just all those daily household activities. But that's fine. There's the systems in place to be able to get care in and support and whatever else. So the human spirit is an amazing thing. We adapt, we learn, we change, and that's where I'm at. I'm finding that I'm accepting where things are at for me, and I've, certainly moving on with my life. And one of the great things that have come out of this for me, and it's really strange. It sort of found me, but I found it, and here we are. But I'm really become a stronger advocate for the MND community as I possibly can. And this is a great opportunity for me to just put my thoughts out there, and how my background, how I'm coping, how I'm seeing the landscape for the world of MND. And I think I'm probably a voice for others who haven't got the opportunity or aren't as vocal as me in terms of how we can change things and turn things around. So that's probably a reasonable summation of my background. So where am I at? I'm doing it, as I mentioned before, I'm doing as much advocacy as I possibly can. And what's driven that is, from my early stage of diagnosis, a frustration for me is that I found that I had to go looking for a lot of things. And I'm so reasonably inquisitive by nature. But as we know, the rabbit hole that is the internet is not a good place to start sometimes. And so I did some research there, but I avoided certain things because there are certain things you just don't want to know. And one of the frustrations was, as I say, that there seemed to be some gaps in the system that I felt, at least, that I didn't have. I shouldn't have been going and searching for things. There should have been more resources available to me and to other people with MND. And it's not just me; it's my family, it's my friends, it's my carers, it's anyone who's connected to me that as I say, the information wasn't readily available. And it's not just the information, it's the supports. And so there are people out there who are in my situation who aren't as resourceful as me, I suppose, and I've just gone and chased things. And they don't know what they don't know. So it's a sort of difficult one to put into words and adequately describe, but I think the bar needs to go a little bit higher in terms of just making it easier for people who are going to be doing it really, really tough for certain periods of their life from here. The other part of that is, and again, it's a sort of a frustration, an observation, but and I'm sure everyone here can completely understand why, but the whole system is set up to support a terminal disease. So we all know what the outcome of that, sadly, is going to be. So it tends to be a reactive environment, I've found, to a large degree, that there's a lack of proactivity. And in my case, you spend the first 50 odd years of your life in a commercial world, as I did before I finished working, and it's all about doing things, and it's all about creating and whatever. And I find it's a difficult mindset shift to move into an environment where things are basically more reactive than proactive. So. And that's across the board. It's me having to ask questions, but I don't know the questions that I should be asking. So it's a really challenging environment. So and I've spoken about this before. What that leads me to is hope. I think that if we use that word as a key word to understand the mindset that people in the MND community who are affected by this, me, friends, family, carers, what they need. And I think if we can really highlight that word and tweak that word up as much as we can and it's, today's a research discussion, which is fantastic. And I'm, really pleased to be involved in it. Research, can we, where is it that we can use the word hope in all of this and just use it as a mechanism for supporting the community that are out there. I just think that's a really, really important part of it all. And hope, means so many things. I don't think it's limited to, it's not right to immediately just think of hope as there's going to be a cure tomorrow. I think we're all, far enough up the path to realise that that's not going to be the case. And I respect there's an enormous amount of work that goes on behind it. But if we can use the word hope in addressing research and addressing findings and addressing what the community needs to hear and understand. So that's probably my key message. I just also wanted to share a few things which, again, I think will help the MND community understand. I use some coping mechanisms which I sort of stumbled upon and sort of found my own way. And interestingly, they the letters. I'm coming out of the banking world. I'm used to acronyms, and here's another one that I found that and the word happy appeared before me. Excuse me, from the mechanisms that I was using. So H for honesty. I think that if we're all honest with ourselves, and I think that's a pretty easy one. And I think the community is really honest in terms of addressing where things are at. But I think from an individual's point of view, the more honest we can be with each other, the more quickly we can sort of move into the right mindset, which leads into acceptance. A for acceptance. Again, accept your situation, understand your situation, understand it's not going to change, understand where you're at, and acceptance of those kind of drivers makes life that bit easier, I think. Perspective for me has been a really, really big one. We've all got our crosses to bear. Mine's different to a lot of others that will be listening to this today, but I'm completely aware that and I see things now, and I'm in environments now physical environments that I've never been before, and so you just see poor people who are it's really struggling. I've been struggling for a long time, and so I am able to create a perspective around where I'm at and my individual situation. So that's been a really important driver for me. Another one is purpose, and this advocacy has been a real strong purpose for me. It's become my biggest purpose. And so having a purpose really, in my case, and I understand don't want to preach about this at all and tell everyone to go out and find a purpose, because we all know life's not that easy. But I think where possible, don't underestimate the importance of having a purpose as an individual and what your role is in supporting the MND community. So as I say, advocacy has become my purpose. I've been doing podcasts. I've been doing some YouTube clips, doing all sorts of things just to get the message out there and try and be a support to the community. Working on another project at the moment, which is a tool to I suppose realign the crossover potentially between MND and mental health. So that's a sort of a work in progress project at the moment. So that's a lot of fun, and as I say, I don't want to underestimate the importance of that purpose. I had to come up with a why to put on the end of the word happy. So it came as why not? Why not be happy? So it was a really simple message for me. So why, as in the letter Y, why not? When you just ask yourself that question, why can't I be happy? And I find it's a decision that you actually make. And I do it all the time. No surprise to anyone. I hate what's going on with my body 24/7. There's not a 5-minute period where I don't get reminded what's going on. And I absolutely hate that. And I think everyone who can see me, would understand that. But I've made a conscious choice to be happy. And that was a why not kind of moment for me to say, and believe me, I've had my meltdowns and early days, but I've been able to rise above that and having this purpose and role has enabled me to really snap into that bracket of happiness and to genuinely enjoy my life, enjoy the people around me, enjoy what I can contribute, and those sorts of things. So again, understand that it is a conscious decision that can be made to be happy and try and turn this thing around. That's probably it for me. I'm not sure how many minutes of our morning I've taken up, but I really appreciate the opportunity to just put my message out there and to really keep supporting the MND community, because as again, I'll come back to this word hope. If I can just really reinforce that word of hope and be a constant reminder that it's really, really important to the people who are more at the more at the coalface of what's going on here. So happy to, I'll stick around obviously for the Q&A. Happy to take any questions there. It's an open book from me. Ask anything you want, and I'll answer openly and honestly. It's my pleasure to introduce Professor Julie Atkin, who's the co-director at Macquarie University Centre for MND Research. Thank you, everyone. Thank you.
Well, thanks, Peter, for the introduction, and thanks for a really great talk, really fascinating insights into living with MND. So what I'm going to do today is talk about our research. So I hope you can see my slide okay. So what I'm going to do today is talk about the basic disease mechanisms and how that relates to this theme of advancing better treatments. And yeah, I work at the Macquarie University MND Centre, so there's about 90 of us working on MND. So we're pretty much wholly focused on MND. So why do we study disease mechanisms? Why are we doing this? What does this tell us about MND? I mean, if you think about your computer or your car, if it stops working, we need to know the cause. So it's pretty much the same as basic scientists. We're trying to figure out what's gone wrong in MND with the idea of developing new treatments. So if we know what causes things to go wrong in the first place, we can do more than just treat the symptoms. And in my group, we try to look for the most earliest things that go wrong in the cell, with the idea that these might be the first things that trigger a whole cascade of events, so that these are the most likely to can be developed for new treatments. And I wanted to mention here, sorry, a study done by AstraZeneca a few years ago. So we've all heard a lot about AstraZeneca, of course, in the last year, because they developed the COVID vaccine, or one of the COVID vaccines. But they actually did a study where they looked at the drug discovery projects, the ones that they'd carried out that had failed, and what was the most important reason for these failures. And it turned out that the most important reason was that the drugs that had failed were the ones where we didn't understand the basic disease mechanisms. So the most important reason then why new drugs don't work is because we don't understand the basic science enough behind the project. So in MND, of course, it's motor neuron cells that are affected. And this is a picture here of a motor neuron. It's a very complex cell compared to other types of cells. So in the image on the right, you can see we've got some red blood cells. And these are much more simple cells compared to a motor neuron that has these very long processes. They've got these long neurites, axons and dendrites, and this is how neurons communicate with each other. And so they're very complex, very large cells, and this in itself creates some challenges in terms of doing basic research to understand what's gone wrong. And when we think about basic science, there's a number of different things that we can look at, but it basically boils down to genes and proteins. So of course, genes give the instructions to make proteins, and proteins are the workhorses of the cell. They carry out all the basic functions in the cell. And I'm going to talk about proteins today because that's the area that my work focuses on. But of course, genes are equally important as well. And what's interesting about proteins is that they form these they produce these very long ribbons, but they produce these very specific structures. So you can see here, this particular protein it's got all these different helical structures. It's got these sheet type structures, and in the middle, in this yellow region, you can see that it forms a very specific structure, and this is very important for a protein because this is how the protein does its normal functions. It depends on the folding of the protein. This is how proteins work. So this is really essential in terms of protein function. Now, what we know in MND is that, unfortunately, these proteins become misfolded. So a protein is produced, as shown here, is this very long ribbon, which initially it's unfolded and then it folds up. If it's correctly folded, it forms this specific structure that, as I've just mentioned here. But what happens in MND is that this protein becomes misfolded. So it forms this aberrant type structure. And unfortunately, this means that it can lose this normal function, and it can also gain toxic or harmful functions. And we can see this in a cell. So this is a particular protein, SOD1, which we know is misfolded in MND, or in some forms of MND. And what we can see in a normal cell is that the protein is produced quite uniformly across the cell. But what we see in MND is that this protein forms these misfolded protein clumps. And we can see this using microscopy. So this is one of the characteristics that we look at in the lab in order to study MND. And why is it green? Well, I just thought I'd show you this kind of cool little tool that we use to study proteins in the lab. We use this particular protein here. It's from a jellyfish called Aquiora victoria, and it's called a green fluorescent protein. And this particular jellyfish lives off the coast of North America, and it's thought that it becomes fluorescent green to scare away predators. And so what we do is use this green protein and we add it to these faulty proteins in MND. We attach it like this so that when we're looking at the cell, we can actually see the protein immediately without having to do any further analysis. We can just look down the microscope and see the presence of these misfolded proteins. But it's really important when studying MND to look at a number of different disease models. So we need to really have a multifaceted approach to studying MND. And that's because each of these different systems, these disease models that we use, have different advantages and disadvantages. So when we think about the proteins and the genes that are aberrant in MND, you know, we can examine this biochemically in the lab. We can also grow motor neurons in what's called tissue culture. These are the sorts of flasks that we grow them in here. And these are great for looking at the fine detail, looking at these proteins or at the genes. But of course, that doesn't tell us what would happen in a whole organism. So of course, you know, the symptoms of MND in patients involve motor impairment and other symptoms. And so we need to be able to see this in a whole organism. So at Macquarie, we use zebrafish and we also use rodent models, primarily mice. And what we do is put these faulty proteins into the zebrafish or the mice, and then we watch how the movement of the animal is affected. And we can track this and identify ways to stop this. And so this is great, of course, but a disadvantage of this is that we can't look in the fine detail using the genes and the proteins. We also look at patient samples, and these are really a really valuable, fantastic source for us to look at, but they also have their disadvantages, particularly post-mortem tissue, because when we look at a post-mortem tissue, we're looking at the disease end stage. So it's really, there's very few motor neurons there, so we can't study those early disease processes. So by using all of these systems together, we're aiming to try and understand what goes wrong in MND. And I haven't got time really to go through all the different things that we know happen in MND at the cellular and biochemical level, but I just wanted to highlight a few things here that these are areas that we've worked on at Macquarie, but also many other teams around the world. So this is a detailed structure of a cell, and I'm not going to go through all the specific details, but I just want to highlight a few types of structures that we look at. So one is the cellular transport system. So if you think, you know, these motor neurons are very long, they've got these very long processes, and so it's really important that they have efficient transport systems. This is so that nutrients can be moved around the cell properly, and also that waste products can be returned back to the cell to be degraded. So this is one system that we know goes wrong at the basic science level. Another one is a structure called the endoplasmic reticulum. This is like the factory of the cell, and it makes proteins, and we know that this becomes clogged up in MND. Another one is the mitochondria. So this is like the powerhouse of the cell. It makes energy. So this also becomes aberrant in MND. And also there's various waste disposal systems in the cell in MND, and we know that there's problems there as well. And finally, the cytoskeleton, which, as the name suggests, it's like a skeleton. It provides structural support. And so we're looking at this as another possible way that we can target and develop new drugs for MND. So this is the sort of drug discovery pipeline that we have at Macquarie. So looking at these basic disease mechanisms, we are trying to identify new drug targets based on these, the basic things that go wrong at the cellular level. And then we put them through this pipeline. So first we look at the protein level, we look at these misfolded proteins, which we go in FLAS, and we've got a high throughput microscopy system that enables us to do that very rapidly. We then identify possible compounds that prevent this misfolding that we see at the cellular level and put them into zebrafish. From this, we then identify compounds that can restore the swimming ability of these zebrafish and put them into mice. And then we look for, to test whether these compounds can increase the survival of these mice and also the paralysis that we see in these animals. And finally, we've got a neurology clinic run by Professor Dominic Rowe, and he's running various clinical trials at Macquarie at the moment. So we've got a number of drugs that are, working their way through this pipeline. My team's got a couple that are showing promise in mouse models, but we're still, this is still a very, very long way towards a new treatment. And why is that? Well, unfortunately, it's the nature of the drug discovery process. It really is like looking for a needle in a haystack. There's many, many compounds that show promise at the basic science level, but unfortunately, they tend to fail along the way, and that's for a whole range of different reasons. It could be due to side effects, poor drug metabolism. They might just simply not work very well when we test them in humans. Or it could be the chemical properties of these compounds. So it's a very difficult process, the whole drug discovery process. It takes a very long time, it can take 12 to 15 years, and it's also very expensive. We've also got the added problem with neurological diseases that we have the blood-brain barrier. So this is a really important structure. It's like a physical barrier, and it protects our brain from pathogens and any other kind of toxins that could harm the brain. But it also basically excludes almost everything except very small molecules. So that's an added challenge for any diseases involving the brain or spinal cord. So that's all really I wanted to say today. I just hope that I've convinced you that understanding basic cellular and biochemical mechanisms is really important when we're trying to understand how motor neurons die, and they're really fundamental in advancing better treatments. So thanks for your attention, and I'd like to introduce the next speaker, who is Associate Professor Rebecca Ahmed. So she's a clinical neurologist at the Royal Prince Alfred Hospital in Sydney, and an NHMRC Early Career Fellow. Over to you, Rebecca.
Great, thanks, Julie, for a fantastic talk. I'll just put my screen on so you can see my slides. So thanks, everyone, for listening today. So what I wanted to talk to you about was biomarkers in what we call amyotrophic lateral sclerosis, or MND. And so I guess the first question is, why do we need biomarkers? And that's because ALS or MND is very complex. It's not a simple disease. And we know that there are a certain degree of patients that just have what we call motor neurone disease at one end of the spectrum. But then there are other patients that develop cognitive changes and have what we call FTD MND that shares both the pathological and genetic overlap with MND. And then we know that at the MND section of the spectrum, there are a whole lot of different diagnoses. So patients can have what we call traditional motor neurone disease, but then they might have flail limb, as Peter mentioned, or they may have primary lateral sclerosis. So it's a complex disease that makes diagnosis and then clinical follow-up very complex. So what we mean by biomarkers, they are by definition, objective, quantifiable characteristics of what's going on in the disease, so the underlying biological process. And we can use biomarkers for a number of things. And over about the last 5 years, biomarkers have really become a hot topic in motor neurone disease, particularly as more and more clinical trials have come into development. So essentially, we can use biomarkers in terms of diagnosis. So they can be a one-off measurement that we can use to diagnose people and say they've got this form of motor neurone disease, or maybe they've got MND with FTD. We can then also use biomarkers potentially as a one-off measurement to try and predict how a patient's disease will progress. So whether they'll be known as what we know slow progressors, so they'll have a slow progression over many years, or fast progressors, where they'll have a quite rapid decline. We can then also use biomarkers as what we call pharmacodynamic, or monitors of disease progression. And that's when we give a person a drug and look at how their biomarkers respond to that drug and their treatment response. There are a large number of biomarkers that are coming into clinical development. And today what I wanted to do is take you through what I think are probably the hot biomarkers for each of these groups. So we know we have biomarkers available for blood and cerebral spinal fluid. So cerebral spinal fluid is the fluid that essentially our brain is and spinal cord is bathed in. We've also got biomarkers related to underlying genetics, biomarkers to metabolic function, imaging biomarkers, and also neurophysiology biomarkers. And the real reason we want to have biomarkers is to understand what's going on in the brain. We think that motor neurone disease essentially begins in the motor cortex of the brain, and then it potentially can spread throughout the brain and into the brainstem and spinal cord and result in motor symptoms and also cognitive symptoms. So when we talk about blood and CSF biomarkers, really the most, hot biomarker at the moment is a biomarker called neurofilament light chain. And essentially this is a protein that makes up the axons, which are essentially almost like the tails of the neurons. And essentially, when a person's neurofilament light chain increases, we take it as a marker of neuronal damage. And it can also predict cognitive change, and also when there's a decrease in a patient's brain volume. In other neurological diseases, such as multiple sclerosis, for example, neurofilament light chain levels have been found to respond to treatment. So there's a hope in motor neurone disease that we'll find a similar pattern. And there are several studies, such as this study here, that shows that across a wide range of what we call neurodegenerative diseases, we can use neurofilament light chain to help diagnose patients with ALS or motor neurone disease having increased neurofilament light chain levels compared to other disorders such as Alzheimer's disease, dementia with Lewy bodies, and some of the conditions that we call Parkinsonian plus, such as PSP and CBS. We also know that neurofilament light chain offers some potential to be what we call a prognostic biomarker, so to predict how a person's disease may progress. And it can be used to differentiate, for example, between ALS and FTD across the motor neurone disease spectrum. What I'm going to do now is move on to genetics, and this as well is a hot area in motor neurone disease. And we know that there are certain mutations that are associated with ALS, particularly the C9orf72 mutation and the SOD1 mutation. And this is where people have multiple generations in their family affected by motor neurone disease or frontotemporal dementia. And it's felt that probably about 50 to 70% of patients with a familial history of ALS FTD, and 15 to 20% of those with what we call sporadic, so there's no family history, may hold a genetic mutation in the C9orf72 mutation. And what we found is that there are markers on the brain that can help predict if a person has an underlying C9orf72 mutation in their genes. And essentially what we found is that these changes in the brain, which are marked in blue, tend to occur in areas that are associated with psychiatric symptoms. And so a lot of patients who hold the C9orf72 mutation may initially present with changes in their mood and also psychotic symptoms and psychiatric symptoms. And it's felt that this may have an overlap with schizophrenia with similar symptoms. The next kind of biomarker that I wanted to talk to you about was what we call biomarkers related to metabolism. And so that's essentially our body's eating behavior, our weight, our cholesterol levels, insulin levels. And increasingly, it's been found that patients with motor neurone disease have changes in their metabolic function. So many patients are what we call hypermetabolic. One of the most common symptoms of early MND is patients lose weight initially. But despite losing weight and maybe not eating as much as they did previously, they tend to have increased cholesterol levels. And this has been found, in fact, to actually improve survival if patients have an increased cholesterol level. We've also found that patients have changes in their insulin levels. And as patients develop cognitive changes, their body mass index may increase. We also have found that patients with motor neurone disease can have changes in their underlying body habitus. So this is one of our patients who, for the first time after he developed motor neurone disease, developed deposition of abdominal fat while he had shrinkage of his arms. And this is a common phenomenon that we see in motor neurone disease, and we think is a marker of the underlying metabolic changes. We know that also as a marker of the underlying metabolic changes, patients can develop changes in their eating behavior. So often patients with motor neurone disease develop a preference for fatty or sugary foods, and this can also affect their underlying cholesterol levels and has been found to be a predictor of improved survival. If a patient can eat more fatty food and push up their cholesterol level, that leads to an improved survival. We know that these changes in metabolism may be linked to a structure in the brain called the hypothalamus, which is a tiny structure, but it pulls a big punch and controls a lot of the metabolic changes that we see in motor neurone disease. And changes in the hypothalamus can be related to changes in weight and also can predict at what age a patient may develop amyotrophic lateral sclerosis or motor neurone disease and how their disease may progress. So what we've defined in our research is really a spectrum of metabolic changes that can help us define the underlying disease processes and how a patient may progress. And we know that these changes involve hypermetabolism, insulin resistance, and changes in cholesterol levels. The next kind of biomarker that I wanted to talk to you about was imaging biomarkers. And so these involve using MRI brains to see if we can predict what kind of disease a patient has, so what their underlying phenotype is, so whether they've just got pure motor neurone disease, or whether they've got motor neurone disease associated with cognitive change or motor neurone disease associated with frontotemporal dementia. And to do that study, we collaborated with a group in the UK. And first of all, what we looked at was changes in behavior. And these circular graphs show you that patients with what we call behavioral frontotemporal dementia have quite prominent behavioral changes. But also patients with the crossover between ALS and FTD, so ALS, FTD, also tend to develop behavioral changes. And also patients with what we would call pure ALS or motor neurone disease, so just motor changes, can have changes in their behavior. Often patients with motor neurone disease can initially present with apathy and lack of initiation, and often it's mistaken for depression, but actually it's part of the disease process. So to look at this, this is quite a complex slide, so I'll try and make it simple, but what we did was look at every structure in the brain and look at how it compared across the ALS FTD spectrum. And what we found was that patients with pure ALS tended to have change in structure called the insula, which is a tiny structure, but that controls both cognition and also a lot of other symptoms such as temperature regulation and autonomic function. And this change was present in other structures of the brain as patients developed FTD as well. So changes in the insula was really a marker of change in motor neurone disease, and may explain some of the different symptoms that we see in motor neurone disease, as a lot of patients develop changes in their temperature regulation, autonomic function, and also metabolism. And so from this study, we've really been able to define the different brain shrinkage patterns that we see in the different disorders, so ALS versus ALS FTD. And so we potentially could use these imaging biomarkers as a way of diagnosing and differentiating patients. With patients with ALS having involvement of the frontal parts of the brain, but also the brain stem. Whereas patients that then go on to develop ALS FTD or FTD at the other end of the spectrum have more pronounced changes in the frontal region and these middle structures of the brain as well. So the last biomarker that I wanted to talk to you about was what we call neurophysiology biomarkers. And this is where essentially we use a technique called transcranial magnetic stimulation. And essentially what that does is pass magnetic fields through the brain to look at how the nerves are excitable. And this has been found to be used extensively in diagnosing patients with ALS or MND having different patterns of changes in their neuronal excitation, and also changes as the disease progresses. And it's now been trialed as a way of looking at how patients respond to different drug treatments. So I'll come back to my first slide. And so I've taken you on a whirlwind of all the kind of hot biomarkers that are available. And as I said, these can be used for both diagnosis, predicting how a patient will progress, and also how they respond to different drug treatments. And essentially the whole reason we do this is to try and develop treatments that can halt motor neurone disease. And essentially a lot of clinical trials at the moment are using biomarkers to see how patients respond to different treatments, and are also collecting blood samples to store to see for biomarkers that we discover in the future. And so I'll thank you for listening, and I'll hand over to our next speaker now, who is Professor Anthony Akari, who is the head of motor neurone disease genetics and therapeutics research at the Perrin Institute. Thank you.
Thanks very much, Rebecca. Hope everybody can see me now. That was a wonderful presentation, and thank you very much, everybody, for the opportunity to talk to you today. I'm going to talk to you about our novel genetic therapeutics in our team, and I'm going to talk to you particularly on the progress of SOD1 suppression therapeutics, the most recent trial in that space, and then come back to some of the work that we're doing more broadly with a novel group of therapies called genetic therapies or antisense therapies. So just a little bit about me, because it's relevant to drug development. I'm the professor of at the Perrin Institute and the Foundation Chair of Industrial Pharmacogenetics, and why I've been given that role is because I've had 18 years of research and development experience in the pharmaceutical industry over in the US, and I've integrated genetics into drug development for various therapy area programs. I've worked at GlaxoSmithKline and Eli Lilly, and most importantly, now working with a startup company which we've based in the US called Black Swan Pharmaceuticals. That company has been specifically set up with my colleagues in the drug development industry so that we can accelerate the development of the therapies that we're building here in Perth, or candidate therapies, and we can accelerate them through the regulatory process and get them as quickly as we can to what we call phase 1 ready to try in patients. It's very difficult to get the therapies that we're developing, or get the interest from pharmaceutical companies. So we've decided to take matters a little bit into our own hands and set this up so that we can start to accelerate therapies. So what are genetic therapies? Well, I'm going to be very specific, and there's a lot of people refer to gene therapies and genetic therapies in the one catch-all, but genetic therapies are very different. They are antisense therapeutics. And what are antisense therapeutics? They are short synthetic nucleic acid analogues, or they're short pieces of DNA-like patches, if you will, that can bind to specific regions, essentially, of genes and turn down some of those underlying toxic proteins that you've been talking about, that you've heard about from the prior speakers, and potentially, we hope, try to change some of those disease mechanisms. So the reason why I came back to Perth is because Perth has been, or my colleagues in Perth here, that is Professors Wilton and Fletcher, have been a pioneer in that space. They've been working in antisense therapeutics for somewhere in the vicinity of 25 years now. And what really has captured my attention is that 3 drugs for Duchenne muscular dystrophy, which are a world first, have come out of that laboratory and have been given accelerated FDA approvals for the treatment of Duchenne muscular dystrophy in children. And these are these antisense therapeutics. Those drugs have now been in children for about 11 years. And I'll come back to this later on through the talk, but have proven themselves to be exceptionally safe in patients over that 11-year period. So some of you may be aware of the tofersen clinical trials, but the therapies that I'm working on, like tofersen, are based on this antisense technology. And again, I'll emphasize the whole reason why I really wanted to come back to Perth and embed in this team is because of the success that they've had in this space. And I think we, our team and others think that at least for some subsets of motor neuron disease, that this kind of technology is the appropriate way forward. But again, I'll emphasize that these are not the only ways that we're going to solve motor neuron disease. This is one technology or therapeutic class, and small molecules and biologicals and all other kinds of classes of therapies have to be taken forward as well. But we think for certain kinds of motor neuron disease, these therapies will be fit for the purpose. And so we're rapidly advancing these therapies because they precisely target the associated genes, and we think that they can slow down or halt progression in some forms of motor neuron disease. So tofersen is what is called a SOD1 suppression therapy, and recently the phase 3 trial for that was completed. So mutations in SOD1 result in an incorrectly produced or misfolded protein, and that protein can aggregate and is toxic to neurons, and that results in ALS. Tofersen is the first of the suppression therapies, and it's targeted to the 2% of patients that carry SOD1 mutations, and it's designed to reduce that toxic mutated protein. So it was trialled in SOD1 patients recently. To much disappointment, it didn't receive or achieve statistical significance in a measure of progression called ALSFRS over a 6-month period. But I think what's important is what the investigators of the study and the designers of the study reported out, and that is that patients who they had expected to decline fast with ALS had a slowing of their decline on this therapy. Now, that may not be statistically significant, but it certainly is a very tantalising and important result to report out. Similarly, patients who had a slower progressing form of ALS appeared to have stabilisation of their clinical functions. That is, those who had a slow progressing form of ALS and have the SOD1 mutations, their disease appeared to stabilise in some cases. So that's a very promising set of results, although met with initial disappointment because the major endpoint of the clinical trial was the ALSFRS score, or a measure of progression. So what are some of the key learnings here? You've heard a little bit about biomarkers as well in the previous talk by Rebecca, which was very nicely sets this up. So what was clear about the tofersen clinical trial? It set out to reduce SOD1 toxic protein, and that's what they were able to show in the cerebrospinal fluid of these patients who were administered the tofersen therapeutic. Most importantly, it showed in that particular biomarker, the neurofilament light chain biomarker, that was reduced in the cerebrospinal fluid of patients. And that is good news. That suggests that, that there's less nerve damage, or that the nerves may be able to repair themselves. And that's at least how I interpret those data. So what are some of the other learnings? Could it be that other measures of ALS progression could be considered for future clinical trials? And, you know, one of the things that we may look at is that because ALS-FRS couldn't show a slowing of progression over the period of time in the tofersen VALOR trial, perhaps other primary endpoints could be used, perhaps biomarkers or patient-reported outcomes. And I think we need to take a look at some different potential primary endpoints going forward in other clinical trials. What's also important is that tofersen was able to suppress the SOD1 toxic protein, but at the higher dose. There was some serious neurological adverse events in about 2 to 3% of the study participants, as reported by the trial. And it's probable that, and this is just my opinion, limits the dose of tofersen that could be used to push the SOD1 suppression higher. So what does that mean? It means that I think if we give tofersen a little bit longer, that we may see, and this is my opinion, that we may see some changes in ALS-FRS over a longer period of time. But what we also need to do is we need to continue therapies such as the one that me and my team are working on, with chemistries that have had a long track record of safety, where we can push the dose of SOD1 suppression higher and get a greater level of suppression. So we need to push those forward, and we and our collaborators are working on doing that. So just a little bit about our group, and I'll come back to our SOD1 suppression therapy. Our group works on genetic markers, and we look to identify those because we think that they're important, that particularly in sporadic motor neuron disease, and if we can find those genetic markers that explain risk or disease course or mechanisms, we can then co-develop those with our antisense specific gene targeting therapies to the correct mechanism, potentially enriching for those patients. That's where we want to go with the work that we're doing. And I think that the tofersen trial is a good example of targeting mutation-positive patients with a specific therapy for that particular kind of disease, or motor neuron disease. And just like many other teams, we have various neuronal cell models that we use to test our antisense therapeutics in, and I'm not going to go into details about that for the presentation today. What I do want to talk about is how rapidly we've been able to advance a number of these antisense therapeutics. The attractiveness of these therapies is that you can quickly build them and optimize them. Our group's been going for about 4 years, and we've been able to develop a number of these therapies in the laboratory. I just want to be very clear and come back to Julie's earlier comment. It takes a long time to go from the preclinical space that we're in, trying to optimize and create and build the rationale and the evidence for these in terms of efficacy and safety. So we have to work through that in a very diligent way. And so we are working, whilst we have a number of therapies here that we're working on, we need to continue to push forward on them in a very cautious way to ensure that they're both safe, and when we go into a phase 1 trial, that they are both safe and have a great chance of being efficacious. So the first of those therapies that we're looking at is SOD1 suppression. SOD1 is a gene that's of particular importance to the Perth team here that I was in. Our team was part of the team in collaboration with Tipu Sadiq that identified that first motor neuron disease gene way back in 1992, and we've always maintained a very strong interest in the SOD1 gene. That we have a SOD1 suppression therapy, as I've mentioned, that is now being readied to go to our first pre-investigational new drug meeting with the FDA. That work has been supported by many funders, and the two of those funders are here. That is both MND in the grants that they've supported us with, and similarly FightMND, that's allowing us to take that through an FDA process, which we hope will ready the drug by October of next year for phase 1 clinical trials. And we have a number of additional drugs here that we're working on. So just a little bit about our SOD1 suppression therapy. We have two drugs that we were working on, and you can see here when we look at the amount of SOD1 protein that we can turn down in a SOD1 mutation positive patient cell line, we can see very effective shutdown with both of our drugs, but drug 2 is just a little bit better. What we were able to show with that in these preclinical models is improvement in the cell's ability to cope with various stresses when we turn it down, an improvement of cell survival, and an increase in the levels of neuron growth markers, which are very promising. And all of these are evidence that suppression drugs could be therapeutic. And that, in combination with the tofersen trial, makes this avenue of approach very promising for mutation-positive patients. But what about patients that don't carry mutations in the SOD1 gene? We think there's a possibility here as well. There's certainly misfolded SOD1 in a number of genetically defined forms of motor neuron disease. And if you look on this slide of brain tissue from patients, the antibody here that stains, or these brown granular pieces here, show misfolded SOD1. So you can see misfolded SOD1 in a whole host of different types of motor neuron disease. And we think that this therapy may be good for not only mutation positive patients, but for some other forms of motor neuron disease as well. But we have to work diligently and prove the science for that. We can't just say that and then just go straight into those patients. So we're trying to build the science behind that so that we can move responsibly into that area. Very excitingly, we have sporadic motor neuron disease patient cells, and you can see here the nucleus, which is a deep blue, stained in those cells. When we put an environmental or a chemical stressor on those cells, you can see here the green misfolded SOD1 that builds up. When we take our SOD1 suppression drug and provide that environmental stress as well, excitingly, we seem to be able to reduce the misfolded SOD1 buildup. And also looking at other cell measures in these patient cells, we can show that the cells seem to do much better under those chemical stresses. So that's a very promising read, but there's a long journey to go there. But I still think that this provides hope for a much broader application for SOD1 suppression therapies. We have many collaborators and there are lots to acknowledge here. I think the most important thing that I want to say to patients here and around the world is that we're a group of researchers, investigators and scientists that care about this disease, that are passionate and that are urgently working towards coming up with paths forward. If we look year to year, perhaps our advancements are incremental, but if you look back over a 5-year period, things have changed significantly. We can now routinely in everybody's lab take patient cells and turn them into motor neurons and test our drugs in patient cells that have been converted into motor neurons. That's a significant change and really is going to change how we can understand this disease in patient cells. So you're not alone on this journey, and we're working urgently around the world and with the good people who have come to give you a talk today. I want to say thank you to all our friends and funders, and this is our team. Now, I'm just going to stop my share, and I'd like to introduce you to Professor Susan Mathers, who's a consultant neurologist at Monash Medical Centre and the clinical director of neurology at Calvary Healthcare in Bethlehem. Thank you, Susan.
Thank you, Anthony. I'm just going to get my slides up here. I just want go first. I apologise, it's just the bar won't go away. Anyway, I might just make a start. I'm just Dr. Susan Mathers. I'm a neurologist and a clinician. And my talk today is really about how do we take research evidence and translate that into good clinical practice to provide better care and treatment for people with motor neuron disease. We obviously are all very keen to see this research evidence translate into the clinic. And we're also equally keen to see that when we see drugs that can work in animal models or cell lines, that we can take these forward into real people who suffer the real human disease of motor neuron disease. I think the thing that I've witnessed over time very much is that people who participate in research in motor neuron disease are not just in receipt of this the goodness of research, but they give back big time. And we've been hearing a little bit about that already this morning. People with MND donate their research data, their clinical information, but they also contribute to all the biospecimens that from blood, urine, CSF, their own tissues and muscle that feed this pipeline of research. And so one can see that it's very much a two-way street. The other thing to say as a clinician is that patients also participate in research about how one delivers care and how one models care for people with this disease. I think it's important to remember that we need to apply the same rigors of research methodology to how we develop better services for people. And Peter's given us a good insight into the gaps that exist even in a first world country like Australia. So the way I think about research is that all of us can have good ideas and come up with good research questions. But we do need to use the rigor of research methodology to make sure that we develop good evidence and that we increase our understanding. And the sorts of things that we've been looking at today that we need to understand better are disease mechanisms, and Julie's talked about those. Clinicians and patients alike are very keen that we can diagnose this disease earlier. It's a miserable time for people waiting in limbo to know whether they've got motor neuron disease or not. And as we can see from what Anthony's been talking about, the earlier we can get on to treating people with this disease and helping them to make decisions about how they're going to manage their lives living with this disease, the better. And so we need these biomarkers that will help us to diagnose MND earlier and more effectively. What I'd like to talk a little bit more about today is how we can use research into improving the care of people with MND. And of course, what's this all about? It's about finding a cure for MND. And so I want to touch on that as well. Wish I could just get rid of this thing and let me open my slides. But anyway, we will keep going. So if you look at all the policy around the world and you look at our own experience, the literature tells us that people with a chronic disease like motor neuron disease that's often progressing quite rapidly benefit from what's called a person-centered approach to care. And that care should come from people across a multidisciplinary team who have some specialist knowledge in managing this disease. So what do we mean by person-centered care, and what sort of questions, research questions, should we be asking ourselves to improve it? the fundamental aspect of person-centered care is that we need to understand and better respect each individual's values, their needs and preferences for care. And I think Peter gave that a very good base for our understanding. He talked about the gaps in information and education around motor neuron disease when he was first diagnosed. And those are time and time again things that patients raise. Where do I go to find the right information that's useful to me? How do I learn about managing this disease for myself and for my family so I'm not feeling, as dependent on others? And how do we communicate about this disease? How do we tell people they've got motor neuron disease? How do we inform them about what that means? How do we educate health professionals to be better at doing this? Clearly, as the disease goes by, new symptoms develop, and we need to be improving the way we manage those and the options that we give people for treatment. MND is one of those diseases that is not just about the person who has the disease. Clearly, it has ripple effects across the whole family and across the friendship groups of these people and the workplace. And many times people want those people to be involved in their care, and they themselves need education around the disease and its management. Access to care is a very important aspect of equality within our society. Does the person living in the middle of the Northern Territory have the same access to specialist care or research, for that matter, as someone living around the corner from my clinic? Well, probably not. And what can we do about that? How can we put forward good evidence to government that health policy might need some reform to make equitable care an option for everyone in Australia? And the old chestnut of how do we join up all the people that get involved in the care of people with motor neuron disease and make that a streamlined process with good communication, the so-called integrated care. And so are we there yet? Well, Mum, probably we're not quite there yet, but we are, I suppose, in Australia, much better placed than many other countries in the world, and we probably need to be grateful for that. What else are we doing in this space? Well, if you want to study a motor neuron disease in Australia, you probably should aim to study the whole MND community in Australia. Good research evidence relies on good data, and that should be population-based data. And that's the idea behind MND registries here and around the world. Many of you may already be part of what is the Motor Neuron Disease Registry. Many of you may have contributed clinical information or blood samples and DNA to the SALSA project, which is a genomic registry. In the last few years, a whole lot of us around Australia, clinicians and researchers and Motor Neuron Disease Australia and Fight MND have been working together to try and bring all of those sources of data together and link them so that we have a very viable, secure and confidential registry for everyone with motor neuron disease in Australia. And those that evidence can also then talk to evidence that's been generated by other communities in Europe and America and other countries that do a lot of research in MND. Because if we can pool large data sets, we're much more likely to be able to crunch the numbers and get the answers that we're looking for. So this data will be clinical data. It will also be genomic data. It will collect evidence on the protein changes and the metabolism changes that you've heard about already this morning, and any other aspects of the disease that come along as research unfolds. It's also linked to lifestyle and environmental data, for instance, because we think they may be important in triggering MND. Importantly, this new database, which is going to be called the MND Clinical Registry, has a patient portal so that each individual person can register themselves in the database. Wherever they live, they don't need to be attending one of the specialist MND clinics. And there they can store their own health information, their care plans, any medication that they've been trialling. And they can also have access to information about motor neuron disease and information about what's happening in research in MND. So that will be a very powerful tool. And we hope that everyone across Australia can get on board so that we can have a very strong whole of MND community voice. Because what can one do with a strong voice? Well, one can ask patients to have input to how we should be developing clinical guidelines, for instance, that can support a best practice care across the whole country. But also that strong MND community voice hopefully can be heard by governments, and that can help change the way we develop health policy and reform it into the future. And so this diagram just sort of shows some of the questions that I think are important. How can we make care and access to research more equitable across Australia? How can we provide better psychological support for people going through diagnosis and living with this disease and their families? We've used a lot of telehealth, particularly during COVID. I think it's got a very important role to play, particularly for people who no longer can travel to clinics or who live a long way away from clinics. The funding for telehealth that allows everybody access to it at the moment does actually run out in the middle of next year, and it's not yet clear whether the government's going to continue to do that. And then there's respiratory care. Of all the treatments that we can offer people in motor neuron disease that will prolong their lives the most, good respiratory care and the use of ventilatory support with BiPAP machines can offer that greatest survival at the moment while we wait for better treatments to come along. But is it readily available to everyone? We talked a bit about information and communication, and Peter highlighted the gaps that he found when he was diagnosed. So how do we make sure that the information that we are developing or providing or having on our web pages for people with motor neuron disease is clear and reliable and easily accessed and used by people themselves? This is a project that I'm running with a number of other collaborators, but in particular Professor Richard Osborne, who is a world leader in measuring health literacy in various diseases and in various countries around the world. He's worked with the World Health Organization. And we've been funded by the MND Research Association, your money at work, to look at this question in the whole MND community in Australia. And so we're surveying people and their families about how they find and use information about MND. And we hope that that will then lead us to being able to develop better ways of doing that. And then lastly, I want to move on to finding the cure for MND. So this is a phase 1 study, and we've heard from Anthony about his drug development leading on to a phase 1 study. Phase 1 studies by their nature are very small studies. They usually only involve a handful of patients, maybe 10 or 12 patients. And that's disappointing for everybody else who might want to be involved in a trial. But why do we do phase 1 studies? Well, we do them primarily to make sure that the drug is going to be safe and tolerable in humans, because we've only up to that point got data from giving it to animals. We also want to know what is the best dose of the drug to take forward into a proper trial. And so we give people different doses of the drug. We escalate the dose making sure it's safe before we move up another stage of the dosing. And once we get to what we think is the maximum tolerated and safe dose, then that's the dose that we'll take forward into a trial. And so this is a trial that's been funded by FightMND and sponsored by Pharmost, which is an Australian drug company, of a drug called monopantal, which has been used for many years in animals. It's a veterinary drug, but we're going to repurpose it in humans because it works in a different way in the human system compared to the animal system. And because we're only looking for about 12 patients, we're only going to do this at 2 sites in the first instance, and that's my clinic in Bethlehem and Macquarie University with Professor Don Rowe. Once we have a drug like this and we've worked out the dose and that we think it's going to be safe to give to humans, or we've got drugs that are already available for other purposes and we know are safe in humans, what can we do to accelerate that process of drug development? And a lot of effort, I think, is now going into making drug trials that bit faster, that bit simpler, and that bit more accessible for more people with this disease. And this is work that's really come from other disease groups as well. These are the kinds of approaches that people working in the field of cancer or other neurological diseases are already using. And so it's called more platform or adaptive methodologies. And it really is in response to a lot of criticism that is out there from people with motor neuron disease saying, you know, that trials are too constrained, they're too restrictive, and they're often too onerous for people who have quite a lot of disability, needing to travel back and forward to clinic to participate. And so the idea of these new methods of running trials are that you can have just one protocol that you get ethics approval for, and lots of contracts put in place and all the paperwork done. You have one placebo group that you compare the drugs to, and then you can have as many arms as you have drugs running at the same time against that one placebo group. So that allows many more people with the disease to be on active treatment. Still gives you a robust placebo comparison of people who are not on a drug. And it allows you to monitor the progress of each arm of the trial. And if one drug is not showing any promise or is being too toxic, you can quickly stop that arm and replace it with another drug and carry on. And drugs that are showing promise, you then can advance them to phase 3 and beyond much more quickly than waiting for those things to be developed and having long gaps between phases of trials. So we in Australia are following other countries in developing this sort of approach to trials. So I hope that in the not too distant future, people will see that there are more avenues for them to be involved in clinical trials if that's what they would want to do. We certainly have seen today that there's still plenty of ways that people can be informed in research activities, both in the primary science and drug development, but also in looking at how we develop better care for people in Australia. So I might finish there. My reflection really on looking back, when I first started in the field of MND, looking after patients maybe 30 years ago, you know, care happened here in the clinic. Research tended to happen over there somewhere, perhaps at a university. At worst, it often happened overseas and didn't come to Australia. And I think what's happened over the years is that we've got much more involved. We've become a much more mixed community of scientists and researchers, clinicians and patients and families. And that we have learned to share our ideas, we've learned to share information and data and the evidence that we can translate into better care. And as we all know, there is a miracle in sharing. I'll pass on now to Dr. Beck Shin, who is the Research Director at Fight MND.
Thanks, Susan. Thanks for another great talk. Thanks to all the speakers today for their presentations, and thanks for inviting me along to speak about funding research in MND in Australia. Just pop up my talk. So my name's Beck Sheehan. I'm the Research Director at Fight MND. My background is in basic research. So I used to work as a laboratory researcher before I joined the foundation 5 years ago. And today I'm going to talk a little bit about how MND organisations are supporting MND research in Australia. So we know the importance of research in the fight against motor neuron disease, but research is really expensive and it does take time. In Australia, MND research receives support from both federal and state government, but a significant amount of the funding comes from Australian MND organisations. So within Australia, there are a number of different MND organisations, many of which you'll be familiar with, MND Australia and the state associations, as well as MND Research Australia, which is the research arm of MND Australia. Then there's also independent MND organisations as well. So Fight MND, MND and Me, which is a Queensland-based foundation that provides support for people with MND in Queensland, and Racing for Mindy, which is in Western Australia. So MND and Me provide some research funding, particularly in the area of fellowship, so supporting a Queensland researcher, and Racing for Mindy is focused on funding research into familial MND in Western Australia. But the two biggest funders are MNDRA and Fight MND. So MNDRA is, as I said, the research arm of MND Australia. It was established in 1984 as a national organisation promoting MND research. They offered their first research grant back in 1988, a $20,000 grant. And they usually commit around $3 million annually into MND research here in Australia, and to date have supported over $40 million into MND research. So Fight MND was founded in 2014 by the three people here impacted by MND. So Pat Cunningham, who lost his wife Angie to MND, Dr. Ian Davis, who lost his battle with MND in 2018, and Neil Danaher, who's had a very public battle with MND. So since 2014, Fight MND has become the largest independent funder of MND research in Australia. Our vision is a world without MND, and we're primarily a fundraising organisation. So we raise money through major campaigns such as the Big Freeze and grassroots fundraising, and we commit around $10 million annually, close to $56 million invested into research. So when we think about MND research, we can think of it like a flow of information. And I think our earlier speakers have done a really fantastic job of introducing this and demonstrating examples of this. So really knowledge from early basic research or even clinical research can then be translated into impact for people living with MND, either through the development of drugs or potential treatments or through improvements in standard of care. And when we talk about drug development specifically, it's often described as a pipeline, which is shown here across the top with the different stages of research, so basic research, discovery research, drug development, and then clinical trials. So when we look at MNDRA and Fight MND, we do focus our funding investments at different stages of the research pipeline. So MNDRA is primarily more focused towards the basic discovery research, whereas Fight MND's focus of our investment is more around drug development and clinical trials. So basic research is that first stage where researchers are really trying to understand the fundamentals of the disease. So asking questions around what's causing the motor neurons to die, and how are they dying, what's different to a healthy motor neurons, and then what roles are genes and proteins playing in this. In basic research, there's an enormous amount of knowledge and understanding of the disease to be discovered, but that next phase is really about how you can translate these findings or this knowledge into benefits for people with MND. So translational research is really sort of finding this idea and then thinking about how to turn it into an effective treatment, or taking a finding from the clinic, taking a finding from the laboratory into the clinic for people with MND. Now, unfortunately, this stage of research, it's often referred to as the valley of death. It's a very challenging part of research because a lot of great ideas or interesting findings will fall down and not translate into meaningful outcomes in the clinic. And this is also a very expensive and high risk part of research. So it's often really difficult to find funding for this area. And this is an area of funding that FightMND is really focused on. And to talk about this it's really been thanks to Dr. Ian Davis's vision. So when Ian was diagnosed, he was a cancer doctor. So he would have people come in diagnosed with cancer, and he would say that he would open the cupboard and there'd be a whole cupboard full of different drugs to trial. And if one didn't work, they could move on to the next. And he couldn't believe over the first couple of years of his diagnosis that not that there wasn't any new treatment available, but also that there just didn't seem to be a lot of drugs in the pipeline. And particularly for Australians, there was just very limited access to be able to participate into clinical trials. So that's really driven the focus of where FightMND invest their funds. So the next stage of research is the clinical phase, where a treatment moves into the clinic for testing in patients. Each of these phases of a clinical trial, so phase 1 through to phase 4, is incredibly expensive. It needs to be done in a timely, there needs to take time to do this process as well, to make sure that we minimize the risk to participants in the trial. And then once a drug moves beyond that phase 2 study, the cost of the trial really becomes enormous, and it just wouldn't be possible without engagement with government and the pharmaceutical industry as well. So not included in this drug development pipeline is, of course, clinical research, where findings in the clinic can be translated into new knowledge and advances in best practice standards of care. So through MND organisations, over $93 million has been invested into MND research, and almost $70 million of this has been in the last 5 years. So between MNDRA, also MND and me, who also fund some research, and FightMND, we fund research through a range of different types of grants shown here. So just more discovery research grants from MNDRA, and more of the clinical type research grants through FightMND. And then the overlap is through fellowships and scholarships, which is funded by both organisations, and also through MND as well. So with this large amount of investment, we have a really strong obligation to ensure that all research that is funded is of an excellent standard. And both organisations have an annual grant round and a rigorous grant review process, which utilises MND experts from across Australia and internationally to review and rank grants to recommend for funding. And if anyone is interested in learning more about this process, I'd be happy to provide some more details on this. Also, with this amount of investment, Gethin and I have been doing a lot of work around reflecting on this, the type of research that we fund. And we know that people with lived experience of MND are really critical and are a central voice to this and this decision making around our investments. So we started last year with looking into this through an MND summit, where we brought together key stakeholders from across the MND research sector, and importantly, people with lived experience as well. And we really talked about the way that funds are invested into research and the type of research that's important to people with lived experience. And then off the back of the summit, or the outcome of the summit, we've formed an MND collective, which is really a group of stakeholders coming together to think about and create a framework for how we can work together better in research. And part of this MND collective, we'll have a lived experience advisory committee to help with guiding our decision making. And so both Gethin and I will be happy to provide some more information around this. So please reach out if you would. Sounds like something that you would like to be involved in. So in summary, through MND organisations supporting research, over $93 million has been invested, with almost $70 million in the last 5 years. And the impact that this funding's been able to deliver includes a better understanding of the disease and how to treat it. Also building capacity in the research sector. So in Australia, we're incredibly lucky to have so many outstanding researchers. And with this funding, we're being able to keep the best and brightest researchers, MND researchers in the field of MND, and working on that, understanding the disease and finding effective treatment. We've also been able to support a pipeline of potential new drugs or treatments. So really solving that problem of not a lot of new compounds or potential treatments coming through the pipeline. We're sort of helping things to progress out of the lab and into the clinic. We've also provided more opportunities to participate in research. And I think Susan gave a really good overview about how all the different types of opportunities there are to participate in research, not just through being a participant in the clinical trial, and also a better understanding of clinical research, which leads to better care interventions, and of course, more access to clinical trials for people to participate in. So I'd like to thank you, and I'll throw back to you, Gethin.
Thank you, Beck, and thank you to all our presenters. I'm sure most of our audience members have their heads spinning after that. That was a real tour de force that took us right through from the patient's perspective, basic research, right through to how we can really improve this. So we have a few questions, and hopefully if your head stops spinning a little bit, you can fire a few more questions into the chat for our audience. First up question, this comes through to for Rebecca. Is there a difference between bulbar onset and other forms of MND, I guess, in terms of biomarkers, etc.?
Yes. So we know probably about 30% of patients will present with what we initially call bulbar onset MND, and that involves symptoms that mainly involve the muscles for swallowing and speech. Usually as the disease progresses, patients will then develop limb symptoms as well, and vice versa. So patients with limb MND can develop bulbar onset MND. We think that there may be a difference between bulbar versus limb onset in terms of cognitive changes. A lot of patients with bulbar onset MND develop what we call a pseudo-bulbar effect. So they have emotional lability, so they'll often cry or laugh inappropriately. So they might see something on the TV that we might laugh for 2 seconds about, but a patient might laugh for a long period of time about. And so at the moment, there are studies looking at, are those differences due to changes in the brain, and are there ways that we can tell the difference and how patients will progress? So that's kind of an area of research at the moment.
And before I sort of charge into all the questions a bit more, Peter, did you want to add anything from your perspective at all after listening to the talks?
No, look, I'm really grateful to be able to sit in on everything, and it's funny, I don't come from a medical background at all, so a lot of it just washes over me. But I really take such a positive outcome from the fact that all this work's being done. And when I do talk about hope, I'm in a enviable position right now, being able to see everything that's being done, and all the the experts in the room who are really throwing their heart and soul into this. So yeah, when I talk about hope, it's evident here today. So I'm really grateful to have a front row seat to that. So but look, not really. I just giggled to myself then about the increase of emotions, and wow, you're describing me here. It's you put me in front of a TV, and then the sad cat stuck up a tree or something, or I'm done, I'm hopeless. So I've got to sort of look away and bite my lip so I don't tear up. But yeah, so sorry, that's just a little aside. But no, look, nothing really come out of it other than the fact that, as I say, it does give you a bounce in your step to see that all this stuff is being done, and there's so many committed people who really, really genuinely taking on the cause. So on behalf of the community, I'm very, very grateful for what I've learned today.
Thank you, Petty, and I did like your happy acronym. It's a fantastic approach, I think, and just represents some of the resilience that most of the population show. It's just amazing that you guys can just carry on like you do, I think.
Yeah, appreciate that. Thank you.
So another question we have, this one's probably for Anthony. People are loving your trials and are wondering if there's any chance that these could be taking place in Adelaide as well.
So the short answer to that is we never know. What we're looking for as a sponsor as we take these therapies and get them through an IND submission ready status, a sponsor that can help take them into phase 1. That's driven by where the sponsor wants to do it. We would hope that it would happen in Australia, but that all focuses on the sponsor and where they would do those sorts of trials, unfortunately.
Okay,
no reason why it couldn't happen in South Australia is what I would say.
Yeah, I guess ideally you'd always want to have a trial that was national, that would have sites in every state in an ideal situation. Another question here, this is kind of more generic question that a few people might want to pitch in for. So is there any research around interactions of daily living that makes MND worse? Coffee causes muscle twitches. Good coffee therefore contribute to make muscle MND progress faster. Some people are allergic to peanuts. Are some people more predisposed to MND because of such things or other common compounds? I know there's been a bit of work in environmental factors out there. Does anyone want to jump in first? Susan?
I'll be happy to take that one. Gethin, thanks. Yeah, look, we've heard a lot today about the genetic underpinning of MND and MND susceptibility in the community. But it's not, genes are not the whole story. We know that 90% of people have sporadic disease and only some of them carry specific genes. So there's something else that's triggering MND, and that clearly has to be something in the environment that we all live. And that might be a lifestyle aspect, or it might be something we come into contact with in our daily lives. So it's always been an area of interest for researchers in MND as to what kind of exposures might trigger the disease. As part of that, you might remember I talked about the registry, the database. SALSA have produced a very large questionnaire that looks at where people were born, where they've lived, what kind of occupations they've done, what other aspects of their lives might have influenced their risk of getting the disease. And so that's actually a survey that's open at the moment, and will be linked to European and other surveys, so that we'll have a huge amount of data on that, because you're going to need a lot of data, because there may be very many environmental factors that can influence the disease.
Does anyone else want to add anything else from there? I mean, Julie, sorry to put you on the spot, but I know, Gilles Guillemin at Macquarie's certainly been looking into environmental factors as well, I think.
Yeah,
sorry for butchering your name, Gilles.
Yeah. So yeah, I mean, I guess, just to add to what Susan said, there's been lots of studies on these sorts of things, on cigarette smoking and exercise, and caffeine's been studied as well in ALS, but there's studies have often been quite conflicting. Some studies have showed, for example, there is a risk with cigarette smoking, and other studies have concluded there's no risk, and some studies have postulated that caffeine might be protective, but other studies have shown not. But yeah, in terms of I guess environment, the BMAA story is, I guess, fairly interesting, and as Gethin mentioned, collaborators at Macquarie are already working on this. So there's an environmental toxin produced by cyanobacteria, which are blue-green bacteria that live in the ocean and rivers. And these accumulate in fish and shellfish. And it seems that they sort of misincorporate into proteins, including the SOD1 that we've heard about today. And this enhances the misfolding mechanism. So it sort of, you know, in terms of a mechanism, that certainly looks interesting, and there's evidence that where these blue-green algae are, there's increased incidence of MND. But I guess in terms of cause and effect, I guess that's, you know, that's a still away from being proven conclusively. But yeah, I mean, it's a very interesting area, the risk of environments and MND.
Thank you, Julie. A quick question back to you, Susan. Is there any, how do you become part of the SALSA program if you're based in Victoria? Does it have to be through a clinic, or can anyone sign up for it?
It's mainly through the clinics, any of the clinics in Australia. I think with the new registry, people will be able to self-register, and then I know that the researchers who are based at the University of Queensland are able to send people out testing kits if they want to submit DNA without attending a clinic. So it's not just for people who can come to clinic. But if you live in Victoria and you don't attend any of the clinics and would like to be part of it, then please, you know, get in touch with us at Bethlehem and we can help you.
Great, thank you, Susan. I have a question here about the MindOz. So again, this might go back to Susan, but how do you see the patient app portal in improving self-management of MND and links with MND associations as well as clinics to help by improving those links? Then how can you provide more responsive, effective treatments?
Yeah, thank you. I mean, I think that question summarizes where we'd like this to go. I think people with MND do want to be as self-sufficient and as in control of their own disease as possible. And so that comes down to information and education, and clearly having a portal is one way of providing some of that. We hope that it will also link them better with the people who are looking after them, so their health professionals, their GP, the service providers like the NDIS, disability providers, may all then have access to the same information if the patient chooses to share their health information with that group. What we do need to have in place is a way of people both anticipating needs into the future, and Peter you really hit the nail on the head with this, because this is a disease about being on the front foot. We do know what sort of things happen down the track in MND, and it's good to think about that and decide for yourself how you would like to handle later stages of the disease, or how you would like to be treated medically if you became sick with the disease. And so anticipating needs is an important part, and helping people to do that, using the right language, having the right educational resources is important. But in the end of the day, you also need someone to respond if you get into trouble or if you have a problem. And if you're well linked to your GP or you're well linked to your clinic, you may already have places that you can go for that advice on the day that you need it. But many people, I think, living with MND in Australia and elsewhere in the world don't have that resource. And I know that people are interested in trying to link that to the MindOz system. How we can do that into the future and fund all of that obviously remains to be worked out.
Thank you, Susan. Question now for Anthony. I was really interested to hear about your proposal, suggestion that perhaps the antisense therapies, say targeting SOD1, might actually be effective across the wider MND population, rather than people just carrying the SOD1 mutations. Could you sort of speculate on that a bit more?
Yeah, so that's not without controversy, but we need to build the science around that. And as Julie has alluded to, normal SOD1 misfolds under certain circumstances as well. And perhaps Julie might comment a little bit further on that. We just think that we need to provide that evidence in animal models. So, for example, we're looking at a fuzz animal model and suppressing SOD1 in that mouse animal model to see if we can change the progression of the disease in that. We've seen some exciting data in sporadic motor neuron disease patient cells where we've induced misfolding. We've been able to suppress that, and the cells appear to be dying less when we do that. That's very early preliminary data, but it's very promising. There's a lot more work to do in that space. So our objective is that if this therapy works in mutation positive patients, can we build the science around the misfolded protein, and could that be something that could be less controversial in terms of going into patients? The thing that we have on our side is that the chemistry that we use is very, very safe because it's been in children for the last 11 years. And so, that could lower the barrier to try this in a certain kind of sporadic patient. But I don't think we can commit to that until we do have more science around it. But I definitely, that's where our mind squarely sits. Now, Julie, did you want to comment on any of that? I don't mind if you disagree at all.
Oh no, I think that's a good point. I mean, yeah, it's very interesting, the idea that in sporadic disease, it's the same proteins that are misfolding. So, SOD and TDP43, FUS, these proteins all misfold in sporadic disease as well as being familial, being mutated in the familial forms of MND. And there's an idea of the six hip hypothesis of MND, where there's 6 separate steps are needed to cause MND. So if you've got a mutation, that might be 3 steps already, or 4 steps, depending on which mutation it is. But it might be that in these sporadic forms, you start with step 1 and then other things happen, environmental steps that get you to the 6 steps. So that's a very interesting idea. I mean, this is becoming more and more, I guess, we're becoming more aware of this. These similar events are occurring in sporadic and familial forms of MND.
Yeah, and I think, sorry.
I was going to ask you a question, Anthony, but you carry on.
So, I think, from my perspective, trying to be purely scientific about this, the question is, if you suppress any of those proteins that are aggregated, you probably will reduce aggregates, but what does that do to the neuron? We need more evidence there. I think the tofersen trial is very tantalizing because for SOD1 mutation positive patients, the reduction of the protein appears to be having a benefit. We need to understand that. My feeling is if these drugs are safe and if we can get them into, if we can show a very reasonable efficacy, I don't think we should set the barrier for efficacy too high or for target engagement too high. So long as the drug is safe, so long as we think that the science is going in that direction, we should go into clinical trials and see if it can change progression in the patient. That's my view on it as well. So sorry, Susan.
That's all right. Can I ask Anthony a question to get in?
Yeah, sure, of course.
Anthony, I mean, I think it is good news, the tofersen results. Clearly, being able to start treatment earlier might have greater benefit. And I know there's interest in using biomarkers to look at when one might start treatment. Did you want to talk a little bit about that?
I actually don't have a lot. I haven't thought about it a lot. In my mind, what's on my mind with respect to SOD1 suppression is if somebody is SOD1 mutation positive and our drug is, or any drug is relatively safe that's shown efficacy as tofersen has, if it's got a nice clean profile, why not even go pre-symptomatic and see if it delays the onset in mutation positive patients? The question for neurologists is the associated myelitis is the benefit risk there? I definitely feel as though with the PMO chemistry that we're working on, because that's what Steve Wilton and Professor Sue Fletcher have developed for the Duchenne kids, that's got a very clean profile, and that makes it a very, very good candidate for pre-symptomatic. You know, and these young folks get it there, but I think I'm speculating too much. I don't know what your thoughts are. Yeah, no, I mean, I was talking to one of my patients just last week, and he knows he carries SOD1. And he said to me, well, the obvious thing is that I should be on the treatment as early as possible. And that makes a lot of sense. And I know that there are trials in ASOs for SOD1 that are actually trying to look at pre-symptomatic treatment. And that's where things like NFL come in, and being able to monitor NFL levels to see if people are developing disease or developing evidence of damage. So I think that is a promising thing for the future. The only thing I'd say about the toxicity of these treatments is we've been running trials of ASOs in Huntington's patients, and there are other groups where yes, there are toxicities. Not all of them seem to have any chronic major trouble. You know, there might be some CSF changes of inflammatory changes, but they may not, in the end of the day, be all that limiting to treatment. So we just need more time, I think, to see. Yeah, I agree. I agree that the toxicity observed with tofersen is not a showstopper in my personal opinion. It may limit the dose, and we might get more suppression with other AO therapeutics. But I think, I'm only expressing a belief now, but I think given enough time, tofersen will show a difference in ALSFRS. That's a belief that I have, that I hold. So yeah, or if you start earlier, before the damage is done, or if you start earlier, which we're going to, hopefully that trial is going to proceed. So yeah.
That ties in really well with a couple of other comments we've had. I mean, Richard's just commented that he'd be very willing to try treatment. Richard's a patient, and he'd be very willing to try treatments that possibly had a higher risk profile, and the hope of getting more effective treatments. And also then we've also got a question about there's unlikely to be a single silver bullet treatment. So obviously probably a cocktail or combination treatment. So are we going to even be looking at knocking down multiple genes? Would you, look at an anti-SOD1, anti-TDP43 or something like that together with other symptom management roles as well?
So the answer is yes. So I think we have to try that. The University of Wollongong, Lisanne Uy, has just been funded by Fight MND to look at some, in her iPSC neuronal cell system, some of the antisense therapies that we developed in the lab, one targeting TDP43 using a different mechanism that's been than what's been published, but also in combination to upregulate one of the genes that's involved in, no, not to upregulate, sorry, my PhD student always corrects me, it's to prevent the downregulation of one of the genes that's associated with neural growth and repair. That combination is being explored in an in vitro cell system in motor neuron disease cells. That'll take a couple of years to get enough data to see whether that's a feasible cocktail or not. But we are squarely thinking about the cocktails. You have to, because it's a complex disease and multiple factors come into play.
Sorry, Beck, did you have a comment?
I was just going to add to that, we've seen in the types of applications we've had over the last few years is a shift towards looking at combination therapies as well, and people looking at new treatments together with existing treatments like Riluzole as well to see what effect that has. So I think it's definitely the way of the future.
Thank you.
Yeah, and Beck, sorry, Beck, the trial, I'm not as familiar with it, but perhaps Susan or Beck, you are, the AMX0035, which is the TUDCA, and I can't remember what else. It's a combination that's showing some promise. Yeah,
definitely. That's a great combination of a couple of different approaches being combined. Question for you back to Rebecca again. Is there any research into how diet may affect or help MND progression?
Yeah, absolutely. So we know from large epidemiological studies that actually patients prior to developing the symptoms of MND exhibit changes in their diet. So they have a strong fat preference. And we think that can carry over onto when they're symptomatic. And we've done studies showing that if you have a higher fat intake, you can increase your cholesterol levels, which can improve your survival. And so there's lots of studies going on around the world that are trialing different combinations of diets, whether it's high fat, high protein, to see how that affects disease progression. I guess the take-home message is the one thing that we don't want patients to do is lose any more weight. So whatever they can do to maintain their weight is beneficial. We also tend to take patients off their anti-cholesterol medication, as there is a lot of studies from multiple groups around the world showing that if you can keep your cholesterol higher, that will improve your survival. So that's kind of a hot area of research.
So, but in your talk, Rebecca, you're indicating sort of different types of perhaps weight loss, weight gain. You indicated that some patients with FTD were actually gaining sort of fat mass maybe, but losing muscle mass. Is that critical as well, though? It's not just about weight, it is what sort of weight as well, is that right?
Yeah, so we're doing studies looking at how body composition can change and how that can affect survival. And it's quite complex because it's also related to metabolic rate. And we know that patients with both ALS, MND and FTD have an increased metabolic rate. And so they develop these eating changes. The hypothesis is to kind of overcome that increased metabolic rate. And then that changes also their body composition. So both patients with FTD and MND develop abdominal fat deposition. And there are studies showing that maybe that abdominal fat deposition may improve their survival. So it's not necessarily a bad thing. And I mean, we're taught traditionally that, you know, the lower the cholesterol, the better to prevent heart disease. But you've got to kind of look what's the most acute issue currently. And that's kind of fighting the MND. So that's why we take patients off statin therapy, encourage them to eat a high fat, high protein diet to maintain their body mass index. I mean, the underlying pathology, we think probably by increasing your lipid intake and affecting your cholesterol levels, that may somehow modify your myelin. So myelin essentially is the lipid coating of your nerves. So there's one hypothesis that by increasing your fat levels, that you kind of protect your nerves, essentially.
Does that cross over into MS as well then? Because that's a big problem in MS, is it?
Yeah, I mean, there aren't many studies looking at MS in that, so I think it's probably separate to that, because MS is more kind of an inflammatory reaction. But then again, that brings into, there is lots of research in MND going on to, could there be an inflammatory reaction that's associated with motor neurone disease, because we know a lot of patients with motor neurone disease also have autoimmune conditions as well.
Great, thank you. I have a question here.
Can I ask?
Sorry, Anthony.
Can I ask? Oh, sorry.
Go ahead.
Can I ask a cheeky scientific question to Rebecca? It's interesting what you're talking about. Rebecca, have you looked at ApoE4 and ApoE2? One of them being a more efficient transporter of, fats, and the other one being less efficient, and how that might interact in MND patients?
No, so we haven't looked at that in MND patients, but that's a good suggestion. And there are studies looking at it in FTD, for example, which we know is linked with MND, showing that that can affect how patients present and how they progress. So, I mean, I think kind of it's complex, and it goes back to kind of the interaction between environment and diet and how that can modify your underlying genetics. And so it's just not going to be a one kind of bullet that solves this. It's going to be multiple approaches that involve drugs, genetics, diet, metabolism, lipid intake.
Great, thank you. We have a question coming here for Peter. What have you found to be the most helpful service or facility or resource for you in dealing with MND?
Oh, that's a good question. I don't think there's a single answer to that, and I think it's probably individual. I think, and this, I can only talk about my experiences, but individual mindset. And the people I've come across, and I've been involved in a couple of groups, group discussions, and those sorts of things. The resilience that I'm seeing is a constant, and yeah, the strength that comes from resilience. So that's probably not an answer because it's an individual thing. But look, MND Victoria, they've been fantastic for me in terms of providing not only the physical supports in terms of what I need in terms of mobility scooter. I've progressed to a power wheelchair now, those sorts of things, but also the emotional support. Just know that there's someone there who's in your corner and who you can talk to, who's sadly seen way too much of this experience. So I really think it's a mental game as much as it is a physical game. So I think an emphasis or an understanding of both those aspects is quite important too. I think if you get on top of it from a mental point of view, then you're probably in a better position to go out and find the physical things that you're going to need. But so sorry, if I don't have a single answer for that question, but that's kind of where I've landed, I suppose. Happy to elaborate if you want.
That's great, Peter. Yeah, as you say, there's no sort of single thing you can do. It's basically just drag in what suits you from multiple resources.
Sure. Yeah. Agreed.
Mm-hmm. Okay. Just a couple of quick questions here. It's probably more for Susan or Rebecca. Are the total hours of BiPAP usage correlated with survival? As in more hours, better survival?
I think the evidence at the moment suggests that early use of BiPAP may be helpful in terms of survival. So not waiting until you really, really need it to start it. There's better evidence for that, I think. The number of hours that people use it probably is a harder thing to study. Although these days, most of the machines will actually record when it's on and when it's not on. And I know that there is a study in Australia that's looking at how we can better manage the use of NIV in the home. And that study is well underway and hopefully will give us a bit more of an insight into questions like that. But I suppose some people use it because they feel more comfortable with it on. So they may not need to use it in terms of their blood oxygen levels all of the time, but they may wear it more of the time because it's just more comfortable to sit and read with it on, for instance. Certainly using it overnight is very useful. And that's when most people start using it.
One commentary I've heard about the non-invasive ventilation is that in some areas or some people perceive it more as perhaps more like palliative care or rather than actual treatment. Do you feel that can be a bit of a barrier to increasing the uptake of NIV, where it, like you said, early on disease, where perhaps it can provide that real benefit?
Yeah, look, I think it's a form of treatment and it's a choice that people can make for themselves. Because it's not invasive, people can try it and put it aside if it's not for them. And not everybody can adapt to it. They don't necessarily like the sensation of using it, but many people do, and it's a strikingly helpful way of managing symptoms caused by respiratory problems in MND. So if you think of anything that improves symptoms as being palliative, yes, it's a very good palliative tool, but it's a palliative tool that makes you live longer as well. So its two pluses are that it helps you sleep much better, so you feel better in the daytime, and it often improves people's appetite and energy levels in the daytime, and it clears their head because it gets rid of all the toxins overnight, and so they wake up with a much clearer head and they can think more clearly. But it also, we know, can prolong life significantly, and people can live an average of maybe 15 months more than they would have done without using NIV.
Great, thank you. Quick question here.
Sorry to jump in, Gethin.
Sorry, Peter. Yeah, please
just
do.
endorsing everything Susan says there. Yeah, I've been using one for probably close to 12 months now, and quite reliant on it when I sleep now. So and everything Susan's saying is pretty much spot on for me. So, but again, sorry to put a negative spin on things, but I speak to people and they don't know it exists. Well, haven't been offered the opportunity. So
yeah.
And
that was the point of my slide. Really, was I'm well aware that that is not equitably spread across Australia, and we need to change that guideline, change that policy.
Sure. Yep. Agreed.
Quick question here. Does anyone know about the Emerald trial with the use of cannabinoid oil? I know it's going on up in the Gold Coast, I think, in Queensland, but I'm not aware of the current progress. Does anyone else have any updates on that? Oh, sorry. Okay. Couple of questions around the cholesterol as well. So what you're suggesting, Rebecca, is that patients who are on statins do need to actually come off those drugs and actually take the hit, I guess, of increasing the levels of the bad cholesterol.
So what I would say is talk to your doctor, because I think it's a decision that you make kind of with your doctor, kind of examining the risk benefits. So, I mean, in most people, if no one had if you didn't have a history of heart disease and you had MND, we would take you off the cholesterol and the cholesterol drugs. And so it's something that I would say discuss with your doctor and kind of weigh up the risks and benefits. But I mean, the trials that we use to justify giving people anti-cholesterol medications for heart disease, it reduces your risk of a heart attack or a stroke over many years. Whereas MND, it's a much more acute situation that you want to improve your survival.
And we have a related comment here from Richard, who says, could you please publish that data so he can convince his GP to take him off the cholesterol, the statins, I think.
Yeah, we have published that data.
Okay.
So maybe you contact MND RA, I can send the papers on to you. Okay,
right. We'll make sure we get those to you, Richard. Just a final question. This is kind of one I'm making up now. It can go around the panel a bit. So obviously myself and Beck, we're quite focused on fundraising and how we can spend money. But, what if, a sort of truly dream scenario might be that we got one of those big mining families who like digging up Australia to actually come in with, say, $100 million rather than 5 or 10. What would we do with that if we had someone kicked in $100 million to spend on MND research in one hit? You can start off, Beck. Sorry to put you on the spot.
It's such a tough question to answer because there's so many different areas that you could invest in. And I think each is equally important. And I think I've really noticed that coming from a basic research background, and then thinking that what we really need is clinical trials. But that, a lot of clinical trials have failed. So that understanding that basic research and the fundamental causes of MND is going to lead to better drugs, and that part of research is really expensive. So I think it would be about making decisions around investing it across the whole pipeline to ensure that you're not just focusing on one area because all are equally as important. So that's not really a one-shot answer, but yeah, that's how I think. Shall I throw to someone next, Julie?
Sure. Yeah, I agree with Beck, actually. I think really research is so underfunded generally. I mean, of course, MNDA and Fight MND are doing a really amazing job raising funding, but we never have enough money to do all the research that we need. And I think, the idea, it's been, I guess, obvious from the situation with the COVID vaccines, how quickly, lots of money was invested and how rapid the progression has been in that that space, I think it really just shows how much we could achieve with more funding. But I think, yeah, I agree across the board. I mean, all of these areas are really integrated in this sort of research pipeline that, you know, we've talked about today. That all of these areas really are all, you know, need investment and funding.
Anyone else want to chuck in an answer for that?
I would.
So all of you. Rebecca first.
I would say I think there needs to be a. I agree with everything else said, but a focus on collaborative research, not just within Australia, but also worldwide, involving longitudinal studies where we harmonise all our protocols. Because often what happens is research is one group finds one thing in one part of the world and a different one in another part of the world, and we don't know which is which. So if we can have longitudinal research that's collaborative across the world, and that takes a lot of money, but that would be, I think, key with the other kind of drug development.
Great idea. Susan.
I would fund your collective, because
Awesome.
I think it's going to be across all of these issues, and it's going to embed the patient's point of view at the heart of it all. That's the philosophy behind the collective. So I think that's where the money should go. It will cover all the bases. I
think $100 million would certainly start the collective off with a bang. Would be great. Anthony.
So 100% agree with all of my colleagues around the table on everything that's been said. I guess I would like to emphasize two things. One of them is that we need continue to support the longitudinal studies that are being done. Susan's touched on those, SALSA and so on. We need prospective validation of the biomarkers. So that sort of work is critical so that we can predict progression for patients, and that helps them plan things. And we can also, in a clinical trial, predict progression and look to see if we can change progression effectively with these biomarkers. So prospective validation of those, but also perpetual clinical trials as well, something not dissimilar to what's happening in Boston, where by setting up an ongoing method to continuously do the clinical trials, switch molecules in and switch molecules out and track people on an ongoing basis, would just give us the lower the cost of doing clinical trials and accelerate that process.
Thank you, Anthony. And Peter, do you have how would you spend the money?
I would carve out at least $10 million to put a smile on everyone's face. As simple as that. I think if you change and it's very simple, right? Ask the question, what's going to make people happy, and sort of turn that mindset around. Stop the fight, stop the negativity. And I'm probably cheating a little bit here because I've been speaking to my friends at MND Victoria and having conversations around some fundraising and for Christmas period and all that sort of stuff. So that's a conversation I've had already. That we're looking the concept of make a wish, I suppose. So I thought that was a really, really good idea, just to give people an outlet, give people something different to do. So simply put a smile on people's faces, and that's just not my face. It's the wider community.
Fantastic idea. Cool. All right, I think we've run a fair bit over time now. So I'd like to give people their Saturdays back. Look, thank you all our presenters for a fantastic overview, as well as obviously a great discussion. And thank you for all our participants for firing in with some great questions there. For those watching, everyone who's registered will get a link to the recording. So please spread that far and wide. I think this is a fantastic session and deserves to be seen by lots and lots of people. Just go to our mndaustralia.org.au/research webpage, and you'll find all the recordings there, including this one. It should be up in 24 hours or so. Also, as part of we're going to have a face-to-face research meeting next year, and we'll also have an MND Connect session in there. That will be both virtual and face-to-face for those people who can get to Brisbane. And also we'll be doing a similar sort of format to this Zoom. We'll also allow people to ask questions live as well if they like, so we can make sure it's truly interactive. So that'll be really exciting. That's going to be the 19th November. No, 9th November, sorry, March next year. So we're really looking forward to that, and hopefully again to see a few people again. So again, that's all for today. Thank you for everyone, and please enjoy the rest of your weekend. See you all.
Thank you.